these embryonal tumors are children's tumors that arise very early during the making of a baby. And we don't quite know when they then manifest themselves during childhood, but they're clearly distinct from adult cancers because we believe they mainly rely on some defects, some gene defects that may have arisen very early. So they not come necessarily for environmental type of interactions. And the asset is trying to study what makes them specific, why do they occur, and if we understand the logic of how they are altering the cell to become malignant and how they are rewiring the natural, let's say, molecular wiring system that each cell has, if we understand that, then, and this is the aim of the consortium, then we'll be able to hopefully sort of intervene in a very delicate way so that we are only correcting the cancer cells and not interfere with the rest of the body. Well, we bring, let's say, a little bit of what is called chemical proteomics expertise, which is trying to match chemical agents, also known as drugs when they are mature, to proteins, which are the targets of drugs. So in this works in several ways. We are screening with different chemical agents for so-called vulnerabilities, so particular weak points that the cells may have. And these vulnerabilities arise because of some genetic, let's say, defect. And so we're looking for drugs that are specifically affecting these childhood tumors and not other types of cells. And our group is trying to do that, with combination therapy, so by looking at different agents at the same time, trying to be very subtle about it because it's a subtle problem. At the same time, we're trying to understand how this works then by looking at what these drugs bind and trying to figure out what the molecular nature of their action is. We are taking mostly drugs that have already been approved for clinical use, so we know they're safe in humans. Some of them may not have been particularly well tested in children, but usually it's a matter of dosage. And so then we are using those, as I say, in combination, because if we were to find a combination that is particularly effective, we know that it would be much easier to find approval because they already have been approved to be used in humans, and so in people. And so this is our approach, and these are not that many drugs that we're trying, but the combinations become, as you can imagine, manifold because they are, you have to combine at different concentrations and so on and so forth. So it's a selected group of drugs. These are sort of known drugs, many drugs that work in cancer, but not only in cancer, and they're used at different dosage in different combinations. Being part of the Asset Consortium gives us access to a lot of interesting people who have expertise that is way beyond our own in our own laboratory, that goes from a pediatrician that know the disease very well and know how they're currently treated, but also have experimental models that we don't have, to mathematicians and biocomputational people who are able to try to take our data and derive general principles and statistical observation that we're not able to do, but also people who have technologies that we don't have access to. And the integration of all of this is a very sort of youthful and very nice community, is actually creating the excitement of being part of this. And we're very happy to be part of this, particularly Branka sort of Radic, the PhD student who is involved in this, looks forward to each and every asset meeting because she meets friends, she meets people with whom she has a common quest, which is to treat these childhood cancers across all over Europe and Israel, and that is great fun. It's a nice community, and I believe that the network will stay on beyond the time of asset. We really would like to understand these cancers better. We would like to know how they're wired, what makes them so specific, and they have something different from these adult cancers that we know. We would like to figure out what is it. And if we understand this, then everything else seems to be a little bit easier. If we are lucky, we're able to find even particular targets that may be attractive for drug development. And if we're very lucky, even particular agents that may be alone or even in combination may actually be so interesting that be worthwhile for a follow-up trials, certainly in some model system. In silico we're doing it, but maybe even in humans eventually.